Yes. All right. So let's go over this and see what you have to pay attention to. I think that from chapter 6 is pretty much everything. So intracellular, extracellular, what is considered intra, what is considered extracellular, the different types of transport, the factors that influence the rate of diffusion, what is osmosis, and of course you have to know hypo, iso, and hypertonic, how homeostasis is maintained. Remember, that refers to the dynamic equilibrium, right, that we have in osmosis and diffusion, okay? Because we have to have that isotonic conditions in plasma. Characteristics of carrier-mediated transport, so those are the ones that require some help. So in simple diffusion, carrier-mediated will be an ion channel, right? Because ions cannot cross freely. They can diffuse, right, from high to low, but they need help. They need a protein channel. And of course, we have facilitated diffusion, which it was the GLUT, or the transporters for glucose. That's the typical example of facilitated diffusion, right? And then we have, of course, active transport, which our number one is sodium-potassium pump. Equilibrium potentials for sodium and potassium. And what that means is, where do we have more of amount of sodium and potassium? And remember, there's more potassium on the inside than on the outside. There's more sodium on the outside than on the inside, right? So that's what establishes this equilibrium potential for potassium and sodium. So that is what gives us a membrane potential, right? That membranes are more negative on the inside than on the outside. And then the different way that cells communicate with each other, right? Synaptic, since we already know nervous system, is a cell-to-cell connection. That means that cells need to be very close to each other. Endocrine, we're going to learn all about it in chapter 11. So that means that hormones are secreted into blood so that they can travel to faraway places, right? So for example, there's a hormone that is secreted from the pituitary, and because it gets into the blood, it can reach the ovaries, right? So it's up here in the brain, but it reaches all the way down to the ovaries. So that's the beauty of the endocrine system. Thyrocrine regulation means that there's a cell that secretes a chemical, and that chemical travels a short distance, not as short as synaptic, so it'd be a little longer space, but it's going to go and have an effect on another cell that is close by. And then receptors. Remember that in this chapter, we talked about receptors that are on the cell membrane and receptors that are in the cytoplasm. So we have extracellular receptors and intracellular receptors, right? Okay, so that's for chapter 6. Chapter 7 is our introduction to neurophysiology. So the different types of neurons, that's easy, mostly anatomy. The myelin sheath, right? The covering of fat tissue. The significance of the blood-brain barrier. That's important because the blood-brain barrier protects our brain from getting anything in there. So mostly astrocytes are the ones that are around blood vessels, and they act as a barrier for certain molecules, like some poisons or things like that, that we need to keep away from the brain, as it's such a sensible type of tissue. This one is important, step by step, how an action potential is produced. And I mentioned this when we did this last week, that I may ask you what is a excitatory postsynaptic potential. or an inhibitory postsynaptic potential, right? So you have to know how the action potential is produced, which is that wave of depolarization traveling through an axon. Characteristic, how is action potentials conducted in unmyelinated or myelinated? Remember, continuous conduction or saltatory conduction. The structure and function of electrical and chemical synapses. So electrical synapses, we don't have that many. They don't have a neurotransmitter in between the two cells or the two neurons. We have some electrical synapses in our special senses. Most of our synapses are chemical, so it means that they involve a neurotransmitter, which is a chemical. Here we have the excitatory and inhibitory postsynaptic potential. So remember EPSP or IPSP for short. And then we talked about the receptors, right? We have the ligand gated, means the typical channel where the acetylcholine attaches and the channel just opens, causing a rush of sodium ions inside. But we also have the other kind, which they're called G protein linked or G protein coupled, right? And the example that we used was the acetylcholine, where it attaches to the G-linked receptor, and then the beta gamma little subunit detaches from it, right? So in this case, we use acetylcholine as an example because we know that acetylcholine can be excitatory, and that is when it attaches to the nicotinic receptor, or it could be inhibitory when it attaches to the muscarinic type of receptor, which is that G-linked protein, right? Receptor. The acetylcholinesterase is the enzyme that breaks down acetylcholine, and acetylcholinesterase enzyme is embedded in the postsynaptic cell. So remember that we need that enzyme because neurons cannot be constantly be stimulated, right? So the enzyme will break down the neurotransmitter, and those little pieces of the neurotransmitter are going to be taken up by the presynaptic, and it's going to be used to make more. So again, here we have EPSPs, excitatory postsynaptic, and then the different types of neurotransmitters that we have, like the monoamine neurotransmitters and how they're inactivated. Remember, we have the MAO or monoamine oxidase inhibitors. That's the MAO is the enzyme, and they're inactivated by the monoamine oxidase enzyme. Dopamine, why is it important? Remember that dopamine is related to Parkinson's disease. When patients don't have enough of the dopamine in those dopaminergic neurons, if they don't produce enough, then they're going to have trouble controlling movements. And then as we have excitatory neurotransmitters, we have to have inhibitory ones, and we have GABA, gamma aminobutyric acid, and we also have glycine as inhibitory. So remember, an inhibitory neurotransmitter will be causing an inhibitory postsynaptic potential. And remember the group of neurons that I drew where they're all kind of like stimulating each other, but at the end, the response is going to be a controlled response. Spatial and temporal summation is spatial means how you can stimulate a neuron in different parts of the axon. So that spatial means space. And temporal summation means when you apply the stimulus within time, unit time. So how often the stimulus is applied to that same neuron. And then I think at the end, there's a little bit of an explanation of long-term potentiation. Potentiation means that neurons not only are going to be stimulated by a neurotransmitter, but there are other chemicals that are not neurotransmitter, but they can kind of like prime the neuron to respond when they're stimulated. So if there's a relationship between not a neuron not being able to be primed to receive the stimulus and depression. All right, and chapter 8, we went through it, so I think, yeah, I think I went over all of this. By regions, distinct functions, yes, lateralization, language. There's a question, is a question on the language centers. So remember that, I already told you 3, right? Question on EPSP, IPSP, and language centers. So at least you should know those. Okay, any questions on the study guide? No? Okay, I'm gonna stop recording.